Good afternoon. Let's start this section because it's a little bit over time. I think people are still very excited from the previous section. I think we need to move on to start this, the last section of today. So welcome to track 4, Science and Technology Innovation. My name is Jing Wen. I'm a lab director at Emory Genetics, and I will be your host for today's last section. As a reminder, this section will be live streamed, and I would also welcome the remote audience. And also remind everyone to download the mobile app, and you can use the live Q&A and submit your question during the presentation. Our Q&A section will be held after the 2 presenters finish their presentation. So our last breakout section will be presented by Steve Gray, Assistant Professor in the Department of Ophthalmology and Gene Therapy Center at University of North Carolina School of Medicine, and Sean Burns, Medical Director at Intella Therapeutics. They will be taking us through the progress of gene therapy and gene editing. Please join me to welcome Stephen and Sean. Steve.
So first of all, I'm so appreciative to be here. It was an honor to be invited to come and speak today at Global Genes. This is the first time, I don't know why, but this is the first time that I've made it out here, and I'm just really blown away by everything that's happened in this meeting, and I'm connecting with so many faces that I've known over the years. It's been amazing. So I've been tasked with trying to give an overview of where gene therapy is at right now. Gene therapy is an area that has gotten a lot of attention in recent years, I think a lot of enthusiasm. And what I wanted to do today is to just walk you through really some information that's more geared to what my focus is nervous system directed gene therapy. I work in a number of mostly pediatric diseases, developing gene therapy approaches, and I hope give you a sense of sort of where the field is right now, what is possible right now, but then also what are some of the limitations. And the thing that I usually will start off saying in something like this is that we need to think about gene therapy as a treatment. We're replacing the gene. We want to think about replacing the gene, fixing the root cause as being a cure. But we really have to think about gene therapy as being a treatment on some spectrum. And it's going to be better and more applicable to some diseases than others. So let me just walk through some of our work about initiation of first-in-human gene therapy for neurological diseases. So I am in the Department of Ophthalmology at UNC. I do have some patent royalties that I've received from Asclepios and Abeona as conflicts. But I'll start off with this introduction, and if you're interested in just getting to understand gene therapy from the very beginning, this is a really good book called The Forever Fix by Ricky Lewis. And one of the things, the promise of gene therapy, of course, is if you fix a genetic disease at the source, if you fix the DNA, then you've permanently solved the problem, hence calling this a forever fix. And that's the hope, and that's the dream, and that's what we're trying to get to. There are a number of ways that you can deliver genes throughout the body. I'm going to walk through, I think, one of the approaches that's become more widely used, and I think has generated a lot of encouraging data. And this is just using this human virus called AAV, adeno-associated virus, and it is something that nobody would ever study normally because it's the only human virus known that doesn't cause any disease. It hangs out. It's sort of a parasite on other viruses. And nobody would care about this except that about 25, 30 years of work have gone into altering this virus so that it can carry other genes in a therapeutic setting. And so you have, let's see, right here, the little arrow right there is pointing to AAV, and those bigger balls are adenovirus. so gene therapy has a rocky history with 1999 when you had a patient that died in a gene therapy trial that kind of put the field on hold. That was with adenovirus, those big balls. AAV is a different virus. And so basically we're scooping out all the viral genes and we're replacing it with other DNA that we want this to carry. It is, I think it's become sort of a vector du jour, if you want to call it that, because there are now over 100 clinical trials that have been initiated with AAV. It's got a very safe track record. And I think I'll show you sort of some of the things that might be possible with this vector. So I'm going to be talking about gene replacement strategies where we're delivering genes, whereas Sean is going to be following up talking about gene editing and how to actually fix the physical DNA inside someone's cells. So I think nervous system-directed gene therapy garnered this really came back on people's radar about 5 years ago or so, and it was really because of this clinical trial. There had been other clinical trials testing AAV vectors for neurological diseases, and some of them may have shown some signs of stabilization or some small things that could be called minor successes. But there was this clinical trial that was actually run out of Taiwan for this disease called AADC deficiency. And if you play the left video, this is what this disease looks like. That this is a gene that is, it has some relations to Parkinson's disease in that there was actually an approach to try to treat Parkinson's disease. This is involved with dopamine synthesis, and so these patients basically don't make dopamine. So they can't control their muscles. It's a really awful disease. So there had been an AAV treatment designed to treat Parkinson's disease to help make dopamine. And this group in Taiwan basically took this gene therapy vector that was being used in this Parkinson's trial and injected it in some of these AADC patients. So if you play the other video, then this is 13 months post-injection. Same patient. So this was published 5 years ago. And since then, this group has been continuing to dose, most of the patients they can find, and the results have been not curative, but they've been pretty spectacular. There's some of these kids, especially the ones that have been treated early, that are going to school and able to live, a somewhat functional life. And this treatment, there's a trial starting in the U.S. doing this, and it's probably going to be an approach that's a little bit better. But this, I think, showed the world what the potential of gene therapy can be. And this disease, this looks amazing. This is a very specialized situation where they could inject this vector directly into a very specific area of the brain and get these specific results. This is not the case for most diseases. Most other neurological diseases, you have to treat the whole brain. But with this, that localized delivery worked. So when I came into this field, and I've been in the field since about 2006, the approach was just what you saw in that trial. You would drill burr holes into the skull, you would insert cannula, and you would just inject the vector directly into the brain. And this approach works really, really well in mice because if you do these injections right, you can fill almost the whole volume of a mouse brain. Now, a mouse brain is about the size of a lima bean. So now scale that up to a human brain, and you can see where the problem is. You're going to be treating little pockets of the brain and really leaving most of it untreated. And this is why a lot of these approaches failed. So when I came in, I and others were exploring ways to try to get broad distribution across the nervous system. This involves intravenous injection of vectors that can cross the blood-brain barrier, or what we've moved to now is doing intrathecal injection into the CSF. Okay, so this is a result that we published this now 6 years ago. You got the result earlier, so this is kind of early in my career of gene therapy. And this is one of those really cool days in the lab, like one of those really, really, really cool days in the lab. Because, we had been using different versions, different flavors of this AAV shell, delivering a gene in mice and just injecting intravenously. And I was I was still doing a lot of work on the bench at that point, and so I'm sectioning mouse brains and I'm staining them, and I had one, and it's the stain where if the gene's expressed, then it'll make it turn black, and all of a sudden all my mouse brain slices turned black, and I was like, oh, well, what did I screw up this time? But then put it on a slide, and then sure enough, these are kind of sections through the mouse brain, and all the black there is where the gene's being expressed. And this is one of those really amazing days when you're a scientist, when you get to run across the hall, grab whoever you can find and say, hey, do you want to be the second person in the world to ever see this? So this really, for me, demonstrated, that this could actually happen, that we could deliver a gene broadly across the nervous system and really treat a disease. So I've been working on this for a good while. There's a number of reasons why we decided to move to injecting into the cerebral spinal fluid. Partly we can drop the dose about 10-fold. There's some other reasons. But we did shift to doing basically lumbar punctures, and we do this in mice. It is as hard as it sounds, but we do it. And then, this was one of those first experiments where, this is looking at spinal cord sections of a mouse coming from the, bottom of the spinal cord on up, and we saw, oh, we're getting a lot of motor neurons here down in the lower spinal cord, and, up at the top it's not quite as impressive, but it is spreading, and we saw a little bit in the brain. And then later we moved the dose higher, and this is a little bit faded, but I hope you can appreciate that now we're getting something similar to what we saw from an IV injection, but we're using a much lower dose, and we're injecting it into the spinal fluid. So it's a way of sort of concentrating the viral vector within the nervous system, the central nervous system. And then just zooming in, these are individual neurons and supporting glia. And so we were excited about the results in mice, and we moved on and we collaborated with Nick Boulis, a neurosurgeon at Emory, and so he did the injections in pigs. These are 15-kilogram farm pigs, so size of a small child. And now in the pigs, we were seeing, 50 to 100% of spinal cord motor neurons across the whole spinal cord. And then we didn't publish this, unfortunately, but we got the brains of these pigs in my lab, and we sectioned them. And now this is kind of the hindbrain, where, again, all the black is where the gene's being expressed. So this is the cerebellum and Purkinje neurons that are involved with balance and coordination. So further excited by that, we did go on and do some studies in monkeys and published this. That's now 4 years ago. Where the conclusions of this is basically you could do intrathecal administration of this AAV9 serotype and get global distribution of the gene across the nervous system. Now, it's not every cell, and it's dose dependent. I think the best that we could probably hope for is something like 20 or 30% of cells, but still pretty good. But again, is this a treatment or is this a cure? And then also there were some barriers that we saw associated with IV injection. We could lower the dose. About a third of you in this room have naturally been infected with AAV, and you have antibodies against AAV9, so if we injected IV, it wouldn't work in you. But if we do it in the spinal fluid, then it will. So this takes me to I think, the real story, which is this disease called giant axonal neuropathy that some of you have heard of it. When I started working on this, we knew of about 24 patients in the world. Started working on this in 2008. People like to call this ALS in kids. It's not exactly accurate, but I think it gets the message. It's fatal around 20 years old. Really brutal disease in kids. And I got involved with this disease that I had never heard of, really because of a family in upstate New York, a family just like a lot of you, that decided that they had a 4-year-old girl that got a diagnosis that they weren't going to accept. And they raised, about $6 million in 6 years. They funneled all of that into research, develop a treatment. And this is what we developed. When we started this, There was no way to deliver a gene broadly across the nervous system. We didn't even know exactly what this gene did, but we knew that it was missing, and we figured if we could replace it, then that would help. So we developed this approach with the AAV9 capsid, the intrathecal injection. We figured a way to package the gene inside this virus because it was a little bit big. And then we went after this disease where we were really trying to target mostly the spinal cord and brainstem, but as we're finding, we want to get it everywhere else also. So we did, this would be about a 2-hour talk for me to go through everything that we did, but we worked in patient skin cells as a proof of concept that replacing the gene would fix them, and it did fix them within days. We did this in iPSC-derived motor neurons from patients, and same thing. And then we have a crappy mouse model for GAN that the darn thing will live forever. So it was a little bit hard to see if our treatment helped it, but eventually we found some things that were wrong with this mouse, and we did improve it. And then importantly, once we started getting thinking about this as something to move forward into a clinical trial, we did a lot of toxicology studies. We farmed those out to a company. So in 120 mice, 14 monkeys, and I don't have it here, about 30 rats, out to about a year post-injection, we saw no signs of toxicity with our approach. So with that, a couple of the evidence that this would work is this is a sciatic nerve of a normal mouse. These are axons, and this is what it would normally look like. That's a GAN mouse, so I think you can appreciate the difference, and this is bad. Although the mice can still run around and, do mousy things even with their sciatic nerves this bad. And then this is a treated mouse. So I think you can visually appreciate that we're doing something good. And then this is just some of our efficacy data. So when we started this, we couldn't find anything wrong with the mice. We have them do this thing called a rotarod, where it looks like a toilet paper roll that rotates, and it gets faster and faster, and you put the mouse on it, and it's like a log roll. You see how long they can stay on it, and then they fall off. So GAN mice can do this okay, but in the green line, you can see as they get older, a mouse lives 24 months. So we have to age them past 1.5 years old, and then they start to have a little bit of a problem with this. And then black is normal mice, so our mice are somewhere in between. So again, this is not a cure, this is a treatment, but we were still, the entire body of evidence that it was something that we felt pretty good about. So now we have the road to an IND. I didn't know what IND stood for. Investigational New Drug. This is an important word for all of you, important acronym. So this is the application to test a new drug to the FDA. So in 3 years, we generated all these good lab results, and we figured out the delivery system, and we packaged the gene the right way. We basically had our treatment. And then in 2012, we went to the FDA, and I'll say our interactions with the FDA were fantastic. They were very compassionate. They bent over backwards to help us. And they understood. When we met with them, we gave the family the first 5 minutes of our 60-minute meeting, and they showed them what GAN was, and they got it. So I know there's a lot of different stories with the FDA. Our stories were good. But now we had 2 more years to do all the toxicology studies, design the clinical trial. We saw some things that gave us concerns about immune responses that eventually we figured out that was okay. And then we went to this alphabet soup of regulatory agencies for another year. My favorite is the RAC because it's named very well. And then finally, 2015, 6.5 years from when we started, we showed up on clinicaltrials.gov. This was exciting for me. And then we hit a milestone in the gene therapy field in May of 2015, with the dosing, first intrathecal administration of a gene therapy drug for any disease. And it was for this disease called giant axonal neuropathy that nobody's ever heard of. And that, to this day, we know of less than 100 people in the world. And it was because of this woman here, Lori Sames, whose daughter Hannah has GAN, and who moved mountains to make this happen. So it's created a belief in me that anything is possible. So where are we right now? We've treated 8 kids, ranging from 6 to 14 years old. We ended up doing this as a dose escalation trial, so we're at a high dose now. It's been well tolerated. And I'll very tentatively say that we've seen a stabilization of motor function, but we really don't have any kind of official trial results. Now, the important thing that I want to stress here is, we developed this approach, and others developed this approach to deliver. We did this to deliver the GAN gene to GAN patients. But once you have the delivery approach, you can put in a different gene for a different disease. And that's what's happening. And that's why gene therapy there's so much happening right now. And you can see here that there's a spinal muscular atrophy clinical trial that was the IV route. It started in 2014. That trial is reporting very good results in 15 infants that by this point would all be dead, and now they're all alive. Our GAN gene therapy trial, there's a Batten CLN6 trial that started in 2016, and now MPS 3A from Abiona, MPS 3A or 3B should be starting, and CLN3, and then we have another project out of our lab for infantile Batten disease that's taking the same approach that we're getting very good results in mice. So, this is just, thinking about this as a platform technology. This is just what my lab is doing, where we have GAN that's in a clinical trial, 3 other diseases that are called IND enabling, which means we're trying to get them into clinical trials, and then some of them that are at other different states. And so this is just my lab. And then we've published on a lot of this, which is good. I'm in academia. And then this is just this is a little bit faded here, but these are just other diseases that I'm involved with through external collaborations. So, all right, cost estimate, and take this with like a huge grain of salt, okay? Because I really didn't even want to put this up here, but I think that it's valuable to at least talk about this. For something like, what I'm doing, and this is just sort of historically what things have cost for us, and the things that I've been involved with, is, proof of concept preclinical, and this is just kind of assuming that everything works great. And then you could do this as like an investigator-initiated sort of an academic effort. Is proof of concept, sort of all the mouse studies. If you have the delivery platform in place, this can be, I think manageable. Toxicology studies, have to do those. Vector manufacture is the big bottleneck, and some of you know this. A lot of vector facilities that I've talked to, they're booking out into 2019 right now, just because there's so much happening with gene therapy. And I think that this is probably a very conservative number. I think it's pushing higher. And then running a small clinical trial just depends on what you're trying to do, okay? But what I'm hoping is that some of these things can eventually get in the range where they could be funded through, say, philanthropic efforts. There's always the NIH that you can try to fund things like this through, but it would be nice if this could be something that could be manageable, that there could be more Lori Sames in the world to drive things forward. So the key points that I want to leave you with is one thing that I haven't touched on yet, and this is a really, 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 really important consideration for gene therapy, is that gene therapy is permanent. You cannot stop taking the pill. If something goes wrong, nobody can do anything about it. So we have to be really careful that when we go in and dose the first patient, that we've got it right. Of course, we're excited because if we do get it right, it's one treatment and it's a forever fix, okay? With current technology, I think for most diseases, we need to think about gene therapy as a treatment and not as a cure. I'd like it to get to the point where it can be a cure, but this is where we are. And I'd say vectors like AAV9 are an exciting platform technology that has now a positive translational track record across many diseases, including human trials. And so with that, I'm part of a group of people that make me look good, and I need to acknowledge them. I have a really dedicated group of people in my lab that spend a lot of hours and think very hard about these problems and spend a lot of time working with mice. And I will say that we're moving my lab to UT Southwestern Medical Center in Dallas, Texas soon, which I hope will be a good opportunity. They'll help me do a lot of the things that I'm doing. And then this doesn't even touch all the different groups and collaborators that I've had, but maybe it's a good start. So with that, I think that kind of ends my talk, and I think we're going to hold questions until the end, but I'm going to let Sean talk about gene editing. Thank you.
All right. Stephen, that was incredible, inspiring story. Thank you for sharing that with me and with everyone here. And thank you guys for your attention, and to the organizers for inviting me. I'm honored to be here. This is my first time at Global Genes meeting as well, and just incredible to hear the stories, and including that last one. So my name's Sean Burns. I'm a MD. I am the medical director of a CRISPR Castine gene editing company called Intellia Therapeutics. If you haven't heard too much about gene editing, that's okay. It's because it's relatively new in terms of think about it as a clinical paradigm. And I'm here to sort of give you an introduction to that. The company is about 3 years old. We've had a lab for about 2.5 years, so our story is still quite young. But I'll share some, I think, promising results that we've had so far, just as a case example. I borrowed this slide from the Global Genes website. So as many of you know in the audience, there's a whole lot of rare diseases out there, and many of them have a genetic cause. And what gene editing and gene therapy as well, really the notion here is that we might be able to address the root cause of these diseases rather than treating the symptoms. So gene editing is, as I mentioned, very recent advance in terms of thinking about a therapeutic modality. The idea here is to go into the gene or genes that are defective in a patient and to correct the mutations that cause those diseases at the source. The company that I work for is focused on a particular technology called CRISPR, but that's not the only approach to this. There are other approaches out there. CRISPR has, I think, gotten people a lot of excited because it seems to be a little bit faster and perhaps easier to use than some of the predecessor technologies, but there's many approaches that could still be useful here. And so just to sort of give a sense of how gene editing compares to gene therapy, and these are generalizations, so I apologize if these seem to be a little bit blunt, but from gene therapy, we usually think about inserting a healthy copy of a gene into a cell that has a defective copy. It's typically delivered using a virus such as AAV, as Stephen was mentioning. The healthy copy is called the transgene, and that can insert into the genome, oftentimes randomly, for example, with a lentiviral approach, or can persist as outside of the human genome as something called an episome, which is generally how AAV works. Gene expression is oftentimes not normal from a gene therapy perspective, so usually a strong promoter is used to try to drive expression of the gene, and that may or may not be dynamic. It may or may not adapt to needs within the cell. in contrast, gene editing, the approach is to correct the gene that's defective at its source, so in the human genome, and not to insert an extra copy from a so-called transgene. This can be delivered by virus, including AAV, the system, or it can be delivered in a non-viral manner. The key, though, is that we want to have a transient delivery approach. So what we want to have is the gene editing machinery to go in and fix the gene and then to go away, so that the human gene is fixed and will continue to function, but the editing machinery is gone. A specific site, namely the endogenous gene, is targeted, and because of that, the gene expression is felt to be to maintain sort of the normal expression. All of the promoter or the enhancers, all the sort of regulatory aspects should not be changed by the editing. So I mentioned there are a few different options for gene editing. CRISPR Cas9 is sort of the newest, and when I say new, I mean about 5 years ago. It was discovered that this bacterial immune system could be repurposed as a gene editing tool. So 5 years is not a long time, but there's now I think it's about 3,000 publications in the last 5 years using this. Before then, there were about like 100 or 200 that talked about it for its bacterial role in immunity. But so there are other approaches. Zinc finger nucleases and TALENs are some of the predecessor technologies. And there are some certain advantages of CRISPR, which I'm not going to spend too much time going into. What is CRISPR? What is Cas9? So CRISPR stands for the system that's this adaptive immunity in bacteria that's been repurposed as a gene editing tool. Cas9 is the protein that's actually sort of the heart of the system. So Cas9 is this protein that you can think of it as a programmable scissors. So it will go and cut somewhere in the DNA that you tell it where to cut. And so the protein is Cas9, and then we provide a guide, which is in the form of RNA, and that tells the scissors where to cut. That's sort of the address. There are advantages of this system compared to some prior systems for gene editing, in that the protein stays constant, so we don't have to engineer new proteins for each target. And the protein is very efficient because it's evolved over millennia to cut very well in DNA. What gives the specificity is the RNA, and then once the protein is in the right location, it will cut both strands of the DNA. We have a double-stranded DNA genome. And then the outcome of that can vary depending on the indication and what you're trying to do. So again, there are some advantages of CRISPR-Cas9. I'm not going to go through this right now, but suffice it to say it's gotten people pretty excited about the application of this for therapeutics. So again, after the cut happens from Cas9, you can tailor the outcome depending on what change you're trying to make to the DNA. So the very basic sort of default outcome is that you can knock out a gene. So you can cut the DNA, and probably most of the time it will repair perfectly, but occasionally it will repair with a mutation. So you can use that to actually knock out a gene that's, for example, causing a disease because it's acquired mutations that are causing problems. So knocking out is very easy to do with the technology. Repairs are tougher to do. You have to provide a template for the cell to know what to repair, how to repair in the way that would be therapeutically relevant. So usually that takes the form of either a single-stranded or double-stranded piece of DNA that you have to get into the cell and then into the nucleus. So it's a little bit trickier to do. There are other applications of CRISPR that I'm not going to talk about at all today, which are not based on cutting the DNA, but actually just using the CRISPR, the Cas9, to get where you want to be, and then actually turning on or turning off genes in a transient manner. That's called CRISPR-I for interference, or CRISPR-A for activation. Those are sort of newer applications, although everything's still less than 5 years old, so it's all new. So where does CRISPR or gene editing fit in sort of the spectrum of therapies? What we think of it in sort of gene therapy approaches share a lot of this sort of advantages here, but as opposed to treating the symptoms, we're really talking about treating the underlying root cause of a genetic disease with curative potential, though, as Stephen said, cure is a strong word. I think what we're talking about right now is treatments that could help us move towards a cure. And so this would be highly personalized. In fact, because we're cutting specific DNA sequences, people with different mutations would have to have different products to really address the individual mutations. Although it could be applied broadly both to rare and non-rare diseases, including scalability. So where do we think about the application of CRISPR? And so there's sort of two main areas, inside the body and outside the body. The first applications for this technology are actually used outside the body, so taking cells out of a patient, modifying them, fixing them, and then putting them back into the patient. And so this is particularly useful for modifying hematopoietic stem cells. So you can take a patient, for example, that has sickle cell disease, try to fix the cause of that in some manner, and then put the stem cells back into a patient and get the red blood cells to no longer sickle. And so a number of companies are interested in that approach for sickle cell disease and other hemoglobinopathies. That's just one example for the ex vivo side. On the in vivo side, we'd actually be trying to edit cells in a person, and so that's much trickier because you're introducing this whole system into a person, and you need it to go to the right cell type and the right tissue and edit and then get out of the body and not cause problems along the way. So that's a higher bar, but those are certainly in the future as well. So what diseases is this applicable for? And it's really, I'd say, I'm putting some examples up here, but these are just examples and not any particular examples that we or other companies are definitely working on or not. This is just meant to be sort of as a proof of principle here. So CRISPR is particularly good at knocking out genes or excising regions of DNA. That's, again, as I sort of mentioned, that's the default outcome for with the technology. So if there's a condition that's a dominant condition, for example, a protein is made that is causing problems, it's very well suited to that, where you can go in and try to knock out that particular gene that's causing the problem. It's also useful if you have to make a genetic change that where the gene or the protein level has to be very fine-tuned, where overexpression would be toxic, for example, or where you need a dynamic effect that really requires the endogenous regulatory machinery. But I put up some examples here for 4 different organs. We're not limited to these organs. You can think of kidney or lung or any other organs that you're interested in, where there's unmet need. It could be useful in those as well. This is just meant as sort of again, just sort of examples that I came up with. So one example that I just want to share some data that we've created at our company is on a condition called TTR-related hereditary amyloidosis. And this is a condition that is, it's a pretty terrible condition, and it's caused by overproduction or production of a form of a protein, a mutant protein in the liver, but it actually causes symptoms in nerves and in the heart. And it affects, while it's a rare disease, it actually affects quite a lot of patients within the rare space. So about 50,000 patients worldwide that are known. It's probably underdiagnosed. It generally affects adults. So I think the median age of diagnosis is somewhere in the 40s. And it's typically fatal within a few years. I think especially those who have the cardiac phenotype, about 2 to 3 years out, most patients are dead. So and the standard of care is minimal right now. There are a few small molecules that are in trials, but have not been approved for this condition in the United States. So our strategy, the company I work for, is to try to knock out this gene in the liver, the one that has the mutations. The reason that we chose this indication as sort of one of our first indications is because we have a great way of getting CRISPR to the liver, and delivery is really key for any of the application of CRISPR. So the approach is to use something called lipid nanoparticles, which are little balls of fat that you can pack things inside of them. These have been optimized for decades for the delivery of something called RNAi or siRNA, which is another modality I didn't mention. So we've sort of borrowed some of that technology to use for delivering CRISPR. And one of the advantages of thinking about this disease as well as a sort of first in vivo program for us is that we can measure TTR very readily in patients to see if we're getting good knockdown. So this is some of the data that we've generated in mice so far, and we've now had similar results in rats and are working on non-human primates or monkeys. with the idea that we could move forward into clinical trials in the coming years. So what we're seeing is that with a single treatment of the CRISPR delivered by the lipid nanoparticles, we can get about a 97% decrease or silencing of the protein in the circulation. And that would be predicted to be therapeutically beneficial. When we look at the whole liver, that if we take the liver out of the animals and measure the editing, we see about 70% editing of the TTR gene. You might say, well, why is it 70% editing but only, but 97% silencing? So the liver in these animals is about 70% hepatocyte and 30% non-hepatocyte. And the editing that we are doing is only in the hepatocytes, most likely. And the hepatocytes are the ones that we're trying to target because of those are the ones that make the protein. So 70% editing of the whole liver may be close to 100% editing of the hepatocytes, which is what we're shooting for. And an important thing, as I mentioned, we're trying to be very quick with the clearance of this of CRISPR and Cas9. So, and also the delivery mechanism, the lipid. So, within about 24 hours, we see I think it's about a 4 log reduction in the circulating levels of the lipid and the mRNA and the guide RNA. And then by 72 hours, it's below the limit of detection. So it's kind of gets in, does what it needs to do, and then it's gone. And we've also looked at, again, this is in mice, we've looked at durability of this effect. And much like Stephen was saying, this is a permanent change. So you really need to make sure you know what you're doing because it's irreversible. That being said, if you do have a positive benefit, it's for life. So at least in these mice, we can see that the editing and the reduction of the protein, the TTR protein, is essentially unchanged out to at least 9 months. And we're conducting a 1-year study. And these are not the same animals at each point. These are different animals, so there's a little bit of bounce along the way. But you can see that really even the animals that were most recently sacrificed have very similar editing and protein reduction as the ones that were sacrificed earlier on. So what are the challenges that remain? And again, just puts this into context, 5 years ago it was discovered this was useful, and this is fast, but we are still, there's a number of challenges that we need to overcome before we're going to be able to treat patients and be successful with that. I think some of them are technical. I've shown you data from mice. I haven't shown you data from rats or from the NHP experiments, but we are moving this forward. But it's really tricky to move across species. And you can really get a lot of things to work very well in mice and even rats, but as you move up, it's much harder. It's not just scale, it's actually biology and changes and differences. So, but we are making those transitions as fast as we can. The big question is, can we achieve the amount of editing that we need or the degree of editing that we need to have the therapeutic benefit? And for each indication, that's going to be different. And so that's going to be certainly a challenge as we move the TTR program ahead, as well as the follow-on programs. Can we identify guides that are very, we call clean, or don't have any off-target effects? So just like a drug could have a side effect, in this case, we want to make sure that the CRISPR doesn't cut somewhere it's not supposed to. And so we spend a lot of time screening a lot of guides to make sure that we find those that are clean and have minimal, if any, off-target effects. And then, of course, we are delivering a protein that was from bacteria. So there's the potential for an immune response in patients who receive this. And while the exposure is limited to 24 hours or 72 hours, we still have to mitigate that if there is any response, figure out if there is going to be, and then how we're going to mitigate it if there is. So that from a regulatory perspective, this technology is, again, brand new. So how do we assess the safety? How do we determine what preclinical data is needed to move into a clinic or to have a successful IND filing with the FDA or CTA filing with the EMA? And the FDA and the EMA and the MHRA, they're new to this too. So everybody's sort of trying to get up to speed at the same time and educate each other. In terms of preclinical animal studies, again, because of our approach is DNA sequence specific, we can't use the same guides in a mouse that we would use in a human. So how do you de-risk the human if you can't use another species? And so we try to find guides that are in common between human and cyno or rhesus monkeys, so then we can at least get some confidence in those species. But it can be really tricky because sometimes the best guide in a human is not doesn't match up any other species. And so how do you deal with that? We don't want to use a guide that is in a species that turns out not to be the best guide in the humans, because of course that would not have the maximal benefit to the patient. And again, because we're so sequence specific, we have to figure out how to have these personalized medicine approaches for super rare patient populations, where we may conduct a trial and there won't be any patients left after the trial because we've treated them and it's a one-time treatment. So how do we approach that from regulatory and other perspectives? And then there's also this public perception challenge. And so CRISPR, for those of you who have heard of it, there's some good and some bad, right? So recently a scientist in Oregon published that he had edited a human embryo using CRISPR. And this gets to ethical and moral discussions about what are the limits of the science, where should we be going or not going, should we be doing so-called germline editing. Just to point out, our company and the other companies in the space have said they're not doing germline editing, but academics and others are moving that ahead. And this idea of a designer baby has come up, which of course we're nowhere. There's so much medical need that we're focused on. This is not something that it's a distraction for us. And then in the popular press, there are concerns this could be used for nefarious purposes. And I put up here this thing about Jennifer Lopez having a show about this, but it's actually on the minds of government agencies as well, that what if a foreign country or a rogue nation tries to use this as a bioweapon? So this is real, and so we want to make sure that we focus the application of the technology on the therapeutic relevance and try to contain these, this sort of discussion as best we can, not let it be a distraction. So we're very focused on moving past through or overcoming these challenges, and we see a lot of potential for CRISPR and for gene editing as well as for gene therapy. Just to mention, I think gene therapy and gene editing are complementary in which diseases and which in their uses and applications. So I think there's sort of space for both, and indications that are more amenable to one or the other. We see this as having a lot of potential, and we're very focused on moving things forward as quickly and safely as possible. And with that, I'll close. Thank you very much.
John, so we have about 10 minutes for the question. Please use the mic.
Thank you so much for that talk. It was very inspiring. I have actually a question for both of you. For gene therapy, I was just wondering, because you talked a lot about gene therapy on certain portions of the brain, and I just wanted to know, are there any gene therapy trials that are happening now where the mutation is expressed in every cell of the brain? And then my CRISPR question was, when, just in your opinion, do you think CRISPR will be ready for mutations that occur in the brain? Because you spoke about the liver, so.
So yes, your question comes, I think, about this treatment versus cure kind of question, because you can have say take GAN, which we're in clinical trials with, as far as your question goes. In that disease, every cell in their body is missing that gene, and every cell in their body is probably affected in one way or another by the loss of that gene. But you have some cells that are more critically affected than others. In that disease, neurons are really, they're badly affected. There are other problems these kids have. And so we're really targeting the therapy with what the technology can do right now to address those most critical targets. Now, if we're very successful in our treatment and these kids start living into their 40s, they may start having other issues. And so at this point, it's just doing the best that the technology allows us to do right now.
And just the question, I think, was about when is CRISPR going to be being useful for the CNS. So I think the key, so there's a couple of challenges there. Certainly one is delivery, and I think CNS is particularly challenging to get to all the cells efficiently. That being said, our first in vivo programs are in the liver, but we're sort of exploring options for delivering outside the liver, but other tissues in the body. CNS is there are a lot of there's a large unmet medical need in the CNS, of course, and some diseases that are particularly well suited to CRISPR, for example, trinucleotide repeat disorders and dominant conditions, where you could imagine going in either just knocking out the mutation, the allele or the copy of the gene that has the mutation, but leaving the other one intact. So I think there's a lot that. So I guess the short version is I don't know when it's going to be ready for the CNS, but I would say that there's a lot of interest in moving into the CNS because it seems like there's a lot of diseases that CRISPR could be good for there. It's just the challenge of getting transient delivery of the system to as many neurons or other cell types, oligodendrocytes, whatever the disease-relevant cell type is, in that space. So it's still early to say when we'd be ready for that, but I can tell you there's a lot of interest in making that happen and pushing it forward.
I was going to say also, I've kind of tried to give you a view of what the technology can do right now. That's sort of been validated, but there is an incredible amount of work around lots of labs, including my own, to make new AAV capsids that can target different types of cells to be able to do something more comprehensive. The technology is evolving very, very fast right now.
Thank you. So I think, okay, you got...
If you don't mind, briefly discuss the mechanism of splicing or scissoring, if you will.
Sure. So this is a bit different than splicing in terms of thinking about RNA splicing, but the bacterial enzyme Cas9, it's a nuclease, an endonuclease, meaning that it's sort of evolved to cut DNA or double-stranded DNA, but in a RNA guide-specific manner. So if you provide the Cas9 enzyme but don't provide a guide, it won't cut DNA. But if you provide a guide, and that guide is complementary, meaning the bases match up with the DNA sequence, then that allows the Cas9 or the scissors to go to the DNA and cut there. And so it cuts both strands. It breaks the bond between the bases and DNA at the same spot. So there's essentially a blunt cut that forms in the DNA. I'm not sure if that's answering your question, but it's the protein only works really if that guide tells it where to go and to cut.
Thank you. So I'm gonna read some of the questions submitted through the app. There are so many we cannot answer every of them, but I will pick some. So the first question is, do either of you have any thoughts on gene therapy working on a child who has lost most of functions due to spinal muscular atrophy?
So I can comment on some of the. So there is a gene therapy trial for spinal muscular atrophy that was being conducted at Nationwide Children's Hospital. I think it's a phase 1 trial that was fully enrolled and it's done. I think that there are plans to continue that trial. In that trial, they did enroll 15 children, infants with spinal muscular atrophy, and the data that's been publicly reported is that it's mostly there are, in my personal opinion, profound benefits that have been seen in those children. And some of them are reaching what has been reported as normal developmental milestones. They do things that an SMA type 1 baby would never do, like sit up, like feed itself. And to my knowledge, all 15 of those babies are still alive, whereas I think most of the children with that disease die by the time they're a year old.
Thank you. And there are two questions regarding beta thalassemia, so we'll ask them together. Have there been studies related to beta thalassemia with CRISPR? And if so, what was the success rate? Were the patient able to continue on without receiving blood transfusion? And then the next, regarding to the same the beta thalassemia question is, what's the difference between targeted beta thalassemia with cell therapy versus gene therapy? What's the best in your opinion?
All right, I will take a crack at that. So in terms of beta thalassemia, so this is a condition where there's not enough beta globin made in the red blood cells, and so there's a chronic anemia. There are gene therapy approaches to that, including a company called Bluebird Bio, which has a lentiviral approach to express beta globin in the red blood cells, and they are conducting clinical trials for that with the published results. In terms of CRISPR approaches, there are a number of companies and academics that have shown some interesting data in preclinical models that you can upregulate fetal hemoglobin, which is a gamma globin, to take the place of beta globin. So I would say there's both approaches are promising for beta thal, and it's still kind of too early to figure out what's going to be the best approach there. Cell therapy versus gene therapy. So the way that I'm not sure if I'm answering the question right, but both the gene editing and gene therapy approaches so far have been to take cells out of a patient, modify them, and put them back into a patient. So it is a cell therapy, but it also has a gene therapy or gene editing component ex vivo. So it's kind of a hybrid model.
Thank you. So we come back to the on-site audience. Any questions?
Great presentations. I had a question about CRISPR, and the example that you gave was around, for the liver example, it seemed like it was basically taking the entire gene out so that it wouldn't be expressed. What about, how do you feel the maturity is of the technology around point mutations and potentially correcting point mutations? And then related to that, for heterozygous conditions versus homozygous conditions? Sure.
So, right. The example I gave was for this TTR amyloid, where you can knock out both copies of the gene, and the patients are fine because the gene is the protein's not required for normal function. But those are, of course, few and far between. Most genes and proteins that we make, we need. For conditions that are dominant and in a dominant way, that either dominant negative or gain of function, where you could just take out one copy. So most genes we have two copies of, two alleles. If the disease is dominant, meaning only one allele is defective, then there may be ways of knocking out just that one gene and doing an allele-specific knockout is the term. And because there's pretty exquisite sensitivity of the guide, of the Cas9 to mismatches in the guide, if you can find a guide that overlaps that mutation site, then you can make it so that it only cuts the mutated allele and not the other healthy allele. So there are approaches to do allele-specific knockout like that. But to actually do a correction of a mutation, a point mutation, it's harder because that gets into, you have to get some sort of a template into the cell to correct that particular mutation. But that's certainly, the technology there is evolving and evolving pretty quickly. And I would say that we and others in this space are working very hard to get that to a point where the efficiency is high enough to be therapeutically relevant. Because some of the other approaches are, again, much more efficient. Right now, we're sort of starting with some of the conditions where we would not have to do these sort of more complicated edits. But those are, we see where that's where we have to go to have the biggest impact. And so, again, every company in the space, as well as academics, are working very hard to really improve the editing. In fact, one way that we and others are talking about delivering the template is using AAV, much like Stephen's using, which is a virus that has evolved to deliver template into the nucleus of cells. So that's one approach.
One question, probably more of a personal question for Stephen. Those of us that have kids with rare diseases do research, and there's usually one or two labs that are specializing in this area. So my daughter has Rett, and so I know you may not know me. So we're very dependent on these people that really like yourself, have taken an early interest in an area. from a research standpoint, gene therapy was dead. Nobody would go into it. Is it exciting now? Are there more labs going into gene therapy and gene editing and being funded so that, we have more shots on goal for these rare diseases?
So I guess I appreciate what you said. I'm not sure exactly what your question is. How's the funding? It's better than it was, but it's still tough. And maybe the little bit of comment that I can make on that from my perspective is that there's 7,000 rare diseases, and there's plenty of common diseases that everybody's focusing on, and the government and everybody else is pouring money into. So in the 7,000 rare diseases, there's a lot more need than there is capacity, and what ends up driving things is when you have patient advocates that make it personal and that make it happen. I would never have touched giant axonal neuropathy. I would never have known that it exists, and nobody would ever care about that disease except that there was a parent that made it happen. And so grab your researchers, hold them accountable, and try to do what you can to support them, but make sure it's the right researchers.
Just to add very quickly to that, I think the regulatory aspect is really important. And so as we think about 21st century cures and Gottlieb as the FDA commissioner, I think that pushing towards designing trials that are adaptive or new paradigms that will allow us to conduct trials on smaller patient populations using intelligent designs, rational designs that get you answers quicker, cheaper, that's going to be huge because part of it is getting the academic labs engaged to really make the key discoveries, and then how do you translate that to biotech or pharma, and you have to have the rest of it in place. And so there's how do you move things forward? Part of it is actually making sure that once you have a proof of principle preclinical data, that you can very quickly get into the clinic and that you can get people excited about this from a pharmaceutical side or biotech side, which I think that has shifted a lot in the last 5 years and continues to shift towards in favor of rare disease research. There's a lot more people focusing on rare disease now than there ever were before from a pharma biotech perspective, and that's because of this paradigm shift, I think, in regulatory aspects.
Thank you, Steve and Sean. That's a very interesting topic and give us a lot of hope for the treatment of rare disease. So this concludes our today's breakout section. Thank you, everyone. So next will be the documentary section, and after that is the backyard barbecue. So please continue to enjoy the rest of the meeting. And thanks again, Steve and Sean.